Hello and welcome to this recorded presentation from Biogelx, which is intended to give a little background information about the peptide hydrogels that we have developed so far and some of the bioinks that we've recently been working on development of for 3D bioprinting applications. So I thought I should start with a little bit of background about the company. So we were spun out from the University of Strathclyde in 2013. At the moment we're 11 employees, 5 of whom are PhD scientists, primarily on the chemistry side of things, as we are sort of approaching this research question from a materials chemistry perspective, although we also have some biology expertise in-house to help us understand the applications that these materials are used for. So as I mentioned, we are 3D biomaterials specialists, and the products that we sell are these synthetic peptide hydrogels. We have patent coverage to use these specific hydrogel compositions, as well as some tripeptide technology, which is still in development, some 3D bioinks, which have been under development recently, and I'll talk a little bit more about today, as well as using these materials for drug delivery applications. So the key benefit of using these materials is that they are of synthetic origin, made up of small peptides, which are naturally inspired but very easy to control, characterise and define the contents of this material compared to some naturally derived alternatives. And the real benefit that lends these materials is that we can very carefully tune both the mechanical and chemical properties of these materials. For those unfamiliar with the concept of 3D cell culture, it's something that we and others recognise to be quite important to the future of cell culture. And really robust cell culture techniques have been very important over recent years, not only in drug discovery, where it allows us to separate cells from the living subject and study them in isolation and their response to exposure to a range of different drug molecules. It can also allow us to screen for toxicology, and it also allows us to do some primary research into disease states that we might be interested in treating in the future. As I mentioned, it's really 3D cell culture that we are interested in. These materials that we provide enable that, and the reason we think that's important is because the majority of the cells in your body exist in a 3D environment, and so it would make sense that we could achieve more predictive results by using cell culture methods which are closer to the physiological environment that your cells would experience. So this extracellular-matrix that your cells would exist in, the ECM, the more we can mimic that environment, the more predictive these cell culture methods should be. As I mentioned, there is a choice of materials out there to perform 3D cell culture, and generally they can be separated into those which come from synthetic or natural origins. All of our products come from synthetic precursors, which allows us to very carefully define the contents of this material, and the benefit of that means that we don't risk introducing unwanted pathogens or growth factors which might affect the behaviour of your cells and the materials. It also improves reproducibility and limits batch-to-batch variation. And finally, if there are aspirations to take your 3D cell culture application into a clinical setting, then really this is quite key to be able to provide that extent of characterisation that you can achieve with synthetic precursors. The precursors themselves look a little bit like this, and really this is the core technology that the majority of the Biogelx products are based around. So it's a mixture of the short peptide FMOC diphenylalanine, or gelator peptide, and the surfactant peptide FMOC serine. The gelator peptide is more hydrophobic in nature, and when you dissolve this in water alone, it forms these fibrous structures that you can see in the AFM image on the right. And this is really to reduce the amount of interaction between the hydrophobic groups on this peptide and the polar solvent, which in this case is water. These fibres are stabilised by pi-pi stacking interactions between the FMOC groups, and unlike the surfactant peptide, FMOC serine, where when you dissolve this up in water, without any FMOC diphenylalanine, the FMOC-S forms these spherical structures similar to micelles, where presumably the greasier part of the molecule, the FMOC group, is hidden from solvent, and the polar group, the side chain, and the free C terminus of the serine can interact freely with water. Now, when you mix these both together and deionize water to make what we term our pre-gel, we get this nice fibrous structure that we'd seen previously with the diphenylalanine, but in this instance, due to the FMOC group, the FMOC-S can co-assemble with this structure to coat this fibre with the polar groups from the side chain and the free C terminus of the FMOC serine. And this is quite important for cell culture applications because it means that these fibres then have a polar surface to which the cells can attach. The products themselves are shipped as lyophilized powders, so a mixture of these small peptides, and the first step to making your gel with these materials is generally to add deionized water to form this pre-gel solution. The cooperative assembly has already begun at this stage, and we start to get fibre formation of the FF and the S. And then upon addition of a source of calcium ions, so calcium chloride solution or normal cell culture media, which generally contains calcium ions as well, we achieve cross-linking of those nanofibres through coordination of the divalent cation. And the material you get, as shown on the right here, is a reasonably robust hydrogel. We study this in-house by rheology. It gives us the sort of results we would expect from a normal hydrogel, and you can see in this TEM image here that it retains that fibrous structure that we talked about. Now, these materials, as I mentioned, are mechanically tunable, and this can be achieved just by modifying the concentration of the peptide precursors used to make the final gel. This is important in cell culture applications because we now understand that the environment around a cell can be quite important to its behaviour, and so it would be important to grow cells from soft tissue in a reasonably soft environment and cells from a harder tissue in a reasonably stiff environment. And so we can tune these materials to make soft, stiff or rigid gels to resemble the environment you might find in, say, the brain or the liver, through to more moderately stiff environments such as muscles and cartilage, and then finally to rigid environments such as bone. And we have a nice application of this tunability here with this publication from a collaborator, Matt Dolby from the University of Glasgow, who has cultured perivascular stem cells using gels of 3 different stiffnesses, and then assessed the expression of markers for neurogenesis, which is the beta-3 tubulin shown in the first graph, chondrogenesis, which is the SOX9 middle graph, and osteogenesis, which is the RUNX2 marker shown here. And with all other conditions being remaining equal, it was found that the stiffness of the gel seemed to affect the differentiation of the stem cells. So in the softer gels, we saw the researchers saw more differentiation towards what we would expect for neurogenesis. In the stiff gels, similar to chondrogenesis, and then in the rigid gels, more similar to osteogenesis. And actually, in a recent webinar hosted by Biogelx, Matt Dolby spoke about some of this research at length. So if that's of interest, then I'd recommend that you go on the website and have a look at that. We also have this nice image which came from this publication in the bottom right, where you can see a single mesenchymal stem cell attached to the fibres of the gel. So clearly the mechanical properties of the gel are quite important to the behaviour of the cells that you wish to grow in it. But one thing that we also have control over using these synthetic precursors is the chemical cues that you might find from the extracellular matrix in nature, and we can mimic that to some extent using our synthetic precursors. So there are several proteins in the ECM which are important to help cell adhesion and proliferation and differentiation. What's beneficial for us is that there's a large background of literature where people have isolated biomimetic peptide sequences from these large proteins to show some of the active peptide sequences involved in these processes. And so we've been able to include synthetic peptides which have been identified and are associated with these proteins, and we can incorporate them into our powders to give improved cell adhesion while using purely synthetic precursors. A recent application of this came from collaborators in the United States who were interested in culturing hepatocytes. And so they performed 3D cell culture using the Biogelx RGD material and compared this to an industry standard in Matrigel. And this is a naturally derived material which is well known for its batch-to-batch variability. And so really, if we could find a good way to replace this material, it would be beneficial to researchers in this area. So after performing 3D cell culture in both materials, using both an immortal cell line, the HepG2 cell line, and primary hepatocytes, in both instances the Biogelx material performed comparably with the Matrigel. After 5 days of culture, it was also found that the hepatocytes grown in the Biogelx material showed similar or superior expression of hepatocyte-specific proteins to those grown in Matrigel. And this was important as this is a sign that the hepatocytes aren't undergoing de-differentiation, which can be an issue when trying to culture these type of cells. Again, some of this work was presented in the recent webinar, so we'll include a link to that in the description of this video. So to summarise the standard range of Biogelx products, they're all from non-animal origins. They all come from these synthetic precursors. They're true hydrogels in that they're greater than 95% water in the final gel. And they're prepared in mild conditions. All the preparation of the gels are done at neutral pH, room temperature or physiological temperature. And the only thing we require to cross-link our gels and achieve gelation is the source of calcium ions. The benefit of having these synthetic precursors is that it gives you the ability to tune the stiffness and the biochemical functionality that's presented on the surface of these materials. And through work with a range of collaborators, we know that these materials are compatible with a wide range of cell types, including, as shown here, primary hepatocytes, fibroblasts, mesenchymal stem cells, and dorsal root ganglia explants. So this moves us on to the area of bioprinting, which I think is quite a natural point to follow on from when you talk about 3D cell culture. There's been a real boom in this research area in recent years. In particular, I think this has been driven by the availability of 3D bioprinters for those with more limited budgets. But I think the idea in general follows on quite naturally, because really this gives us an opportunity to deposit our 3D cell culture material laden with cells in a more accurate manner. The most common methods are similar to shown in the video, these extrusion-based techniques, where essentially you load a syringe body with your bioink, and cells can be contained within that bioink, and either through pneumatic pressure or some other sort of pressure you can extrude the material from a print head here. As before, there's a range of synthetic and naturally derived options when you're looking at bioinks in this area. However, we feel there really is space for a biocompatible synthetic bioink that would be useful for 3D bioprinting applications. However, based on our standard technology, this did require a little bit of extra formulation to convert the hydrogels that we sell commercially for 3D cell culture into bioprintable materials. I'd like to run through an example project of bioink development with you now. So there are a few challenges that must be considered in the development of a bioink. the first being printability. And really the key considerations here are the viscosity of the material, and this helps with the way you can print layers of bioink on top of one another without too much blending, and while maintaining the ability to bear its own weight. The gelation method, as I've mentioned before, for us this means addition of cell culture media, and the final stiffness of the bioink, as we know, is quite important to the behaviour of the cells contained within these 3D materials. We test this in-house using a rheometer and an extrusion printer, where we print scaffolds into air, and then we can add cell media afterwards to achieve the final stiffness of the material. Secondly, biocompatibility is a key concern, because these materials, the cells contained within them should be able to proliferate and be viable. And at the moment we are undergoing a sort of beta testing program with a range of collaborators in academia and industry alike, who are testing these materials with the cell lines that they're interested in researching. Finally, these materials should show good biomimicry, and essentially we can already achieve this in our standard range of products with the inclusion of these biomimetic peptide sequences. So this is something that we're taking forward into the bioinks as well. So when you're looking to formulate a bioink, one of the common approaches is to find either a natural or synthetic polymeric material with a high aspect ratio. The idea being that these long, thin chains will show some resistance to tumbling over end on end and increase the viscosity in that manner. Common bioinks can be based on collagen, so sort of protein-based additives. Commonly you would see gelatin bioinks and derivatives where the gelatin's been synthetically modified to allow things like UV cross-linking. They could also be based on polysaccharides, so agarose or alginate-based bioinks, or hyaluronic acid. And again, these often come in forms which have been synthetically modified as well. However, both of these additives primarily come from natural origins rather than synthetic. Finally, we have synthetic additives. Most commonly these would be polyethers, such as polyethylene glycol, shown here. And while these aren't famed for having great biocompatibility, there are some modified synthetic polymers which can be used as bioinks, which show reasonable biocompatibility while maintaining that synthetic origin. That's quite important to us. During in-house development of these bioinks, the first thing that we look to optimise for is the viscosity, which is something that we can assess by rheology. And we set ourselves this window of 300 to 600 centipoise, with the lower limit being one which gave us reasonable viscosity for the shapes that we printed to bear their own weight and allow them to be printed in layers over each other without spreading too much. And then the upper limit is really to try and reduce the amount of back pressure you would need to apply to extrude the material from the print head. So this limits the stress that the cells contained in the bioink are put under. While we're talking about the stress that the cells are put under, one of the nice properties of the Biogelx materials is they've all been shown to be shear thinning materials, which means under increased shear strain they actually reduce in viscosity. So in some way that can help to protect the cells contained within the bioink. Another test that we do is just an extrusion test, where we apply pressure to the back of the cartridge and watch as it extrudes from the print head whether it forms drops or filaments. And it's quite important for bioprinting application that you can get nice smooth filaments to ensure that you'll have reproducibility. If you want to print the same structure into multiple well plates, then you want to have confidence that that structure is going to come out the same in each well plate to give you results which are comparable. And then finally we also test for print resolution, and we do this just by printing straight lines initially and assessing the spreading of these, and then by printing finer structures such as those shown on the right, maybe a lattice or this nose design shown here, which can really demonstrate the ability to print layers of bioink on top of each other without undergoing too much spreading. So I'd like to work through a quick case study of a recent Bioink development program that we undertook. As I mentioned, we focus primarily on printability in this in-house work on quite a small scale, just making up 1 ml of each formulation at a time. And really the 3 variables that we look to optimise for are the concentration of the peptide, so this FFX variable. We also add in some cell culture media at this stage in Bioink development, so the concentration of the DMEM, the media that we use in this instance. And then finally the concentration of the polymer additive, which we've added primarily to get better filament formation and printability regarding that aspect. The next step then is to perform rheology on these samples. So we really look to optimise that viscosity to get into the 300 to 600 centipoise range. And we also perform a filament test, as I discussed previously, where we extrude from the print head. And you can see here that we have a nice filament forming from this example. After that, we perform a print resolution test, which is maybe quite difficult to see in this slide, but essentially we just print straight lines and then measure using some image manipulation software the diameter of these lines at various points throughout the print. The benefit of working on a small scale means that we can test a lot of formulations in a reasonably short amount of time. And in this example, I think it was 14 or 15 formulations that we were able to assess in the space of a week or two. And highlighted in this graph here is the green window of 300 to 600 centipoise, which we were aiming for. The starred bars are formulations of particular interest, which either fell within that viscosity range or came close to, and also showed good results in the filament and printability test. The most promising formulations then were scaled up, and we were able to repeat these measurements in triplicate to give us some confidence that these were real results. And in this instance, you can see the first bar on both the viscosity and the print resolution test give the best results. The dashed line in the print resolution test was a peg-based bioink, which was used as a standard to compare the line widths to. And so that was the formulation that we eventually settled on, gave us this nice ear structure when we printed that to plan, and you can see that many of the details have been reproduced quite accurately here. And then, as I mentioned, we do some of these finer prints. There's a short video of a couple of them in action, where you can see the bioink stacking on top of each other quite well, especially in the ear print on the right there, and in the lattice in the video on the left. And so that takes us up to date really with the Bioink development. We've so far developed printable formulations of the standard Bioink, as well as the RGD functionalized and the collagen biomimetic peptide functionalized hydrogels. At the moment, beta testing of these materials is underway with a range of our collaborators, and we're on track at the moment for product launch at the start of 2019. As you probably saw during the formulation slides, during this optimization program, we come across a range of formulations at various viscosities and final stiffnesses, and we're very aware that the application that people might want to apply these bioinks to could be varied. And so we will also look to launch this bioink formulation service around the same time at the start of next year, where we can formulate bespoke bioinks to specific research application needs. And so that's something to look out for in the next year. So all that remains is just to acknowledge and thank the team here at Biogelx, our academic collaborators who have helped supply some of the data shown here, the beta testers who are currently involved in the Bioink development project, And of course, thanks for your attention to this video. If you have any questions, then you can reach me here at chris.allen@biogelx.com. And as I mentioned, please check out the website, and in particular the case studies, to see some of the published work using these materials.